Assalamu alaikum. Today our topic is bacterial corneal ulcer, but before starting the topic I would like that you must know about the structure of the cornea, and I would like to show you the structure of the cornea as it is showed on the screen, that it is basically made up of 5 layers. First is the epithelium, then comes the basement membrane, then stroma, and then Descemet's membrane, and then there is endothelium. You can clearly differentiate that the stroma is the widest layer and the epithelium is the outermost layer. It means it is basically exposed to our environment, or mostly exposed to our environment. Now let us move toward our major topic, which is bacterial corneal ulcer. As its name indicates, that it is a sort of corneal ulcer that is caused by bacteria. Now what is an ulcer? An ulcer is any excavated defect that is produced at any particular area as a result of any inflammatory process. Now start its causes. Let us know about the causes, that what are the causes of the bacterial corneal ulcer. As its name indicates, that it is caused by bacteria, but which bacteria are involved here? As they are also written on the screen, that these are S. aureus, Streptococcus pneumoniae, Gonococcus, Moraxella, Pseudomonas aeruginosa, Klebsiella, and Proteus. Sorry. Whenever S. aureus and Streptococcus are involved, then there would be white colored opacity seen. But whenever Pseudomonas aeruginosa is involved, green colored opacity can be seen in the cornea. Then I would like to mention some bacteria here which are not written in this book, that they can even invade your intact corneal epithelium. All these bacteria which are mentioned on the screen can invade only whenever there is any injury to the epithelium or any other outer layers of the cornea. As you can suppose that there is a space created for them, so they can easily invade your cornea. But some bacteria like Neisseria gonorrhea, Neisseria meningitidis, and Corynebacterium diphtheria are those which can invade even your intact epithelium. You can add them up here. Then we will talk about the pathogenesis. First stage in pathogenesis is infiltrative stage. You must know that whenever there is any injury in our body, an inflammatory process starts in our body. There is recruitment of leukocytes, monocytes, neutrophils at the injured site, and they will try to cover up that particular defect produced or that injured area. But it can also become a silent killer. Inflammatory process can also become a silent killer, though it is a protective mechanism. It becomes a killer when this process prolongs and there is necrosis and scarring of that area. And whenever there is scar formation, or whenever there is any fibrosis of any area, there is no going back. That area becomes permanently dysfunctional. So let us start from infiltrative stage. See that, let us suppose that any person's corneal epithelium is destroyed, as it is also an injury. So inflammatory process starts and there is infiltration of polymorphonuclear leukocytes into the epithelium and stroma of the cornea. And at this stage, cornea is seen as yellowish or whitish colored opacity. Then comes active stage. In active stage, what happens, that particular cornea which was involved in infiltrative stage, or that particular corneal epithelium where there is an injury, that area is sloughed off, or it is necrosed. You can suppose that whenever there is epithelium necrosis, like this was your epithelium which was made up of stratified squamous epithelial cells, but whenever this sort of epithelium is necrosed, such type of epithelium is seen, and ultimately a time will come when there is any excavated defect produced here, which is known as ulcer. Here ulcer will form, and whenever ulcer will form, it would start releasing chemical mediators, and sometimes when aggression of corneal ulcer occurs, there is outpouring of those infiltrative cells into the anterior chamber, and this condition is known as hypopyon. Like this, you can see here that this is your anterior chamber. Whenever any defect is produced in this cornea, its epithelium is completely destroyed, infiltrative cells can come into this anterior chamber, and this condition is known as hypopyon. But this is sterile hypopyon. Sterile hypopyon means that there are no bacteria in this chamber. Now, why there are no bacteria? Although it is bacterial corneal ulcer, there are no bacteria because bacteria don't have the ability to cross this Descemet's membrane. So, as bacteria cannot cross this Descemet's membrane, how they can come into the anterior chamber? So, we will see sterile hypopaeon in this case. You can see here, this is hypopaeon. In the inferior third of the cornea, there is accumulation of infiltrative cells. This is known as hypopaeon. You can clearly differentiate here. Now let us proceed towards the next stage, which is regressive stage. You know that as its name indicates that it is a protective stage. Whenever natural host mechanisms start or when we start the treatment, obviously there is enhancement of the normal host response, and as a result, around the ulcer, like shown in this diagram, around the ulcer there is a ring formation of leukocytes, and the remaining cornea will become completely clear. So that is why it is named as regressive stage, as it is protective. All the cornea becomes clear and only a single ring of leukocytes will form around the corneal ulcer. Next comes cicatrization stage. In this stage, healing occurs by progressive epithelialization. Whenever only there is epithelial destruction, then in this stage epithelialization starts. And if there is prolonged or if there is any deep defect produced, then there would be scarring. And I said before that scarring is a very dangerous phenomenon, that particular area becomes non-functional. So here he says that scarring depends upon the depth and size of the ulcer. That what was the size of the ulcer, depending upon that scar will form. Now let us talk about the clinical features. What are the clinical features? Symptoms. You will see that their patient presents with pain. Why there is pain? You must remember or cram all these three modalities which cause the pain. First is exposure and stimulation of corneal nerves, then spasm of ciliary body, and then raised intraocular pressure. You must remember all of this because our treatment will depend upon these modalities. And as you know that cornea is the only area from where light passes and it is involved in our vision. So whenever there is any defect, then patient presents with blurred vision. Lacrimation can also occur because the cornea is supported in front just by tear film. Whenever there is any defect, that tear film would be destroyed. So there would be reflex over secretion of tears, due to which lacrimation occurs. Photophobic patient can also come. Redness of eye occurs, and halos can also be seen due to the corneal edema. Now let us talk about the signs. What are the signs? You must know that signs are those features that a doctor sees in the patient, and symptoms are those features which a patient would tell you by himself, that he has those symptoms like pain, he has blurred vision, he has lacrimation, photophobia, redness, etc. Now signs, what would you see? As I told before that why vision is reduced, you would see that patient's vision is reduced when you would do visual acuity test. Then other conditions you will see are lid edema, that lids would become edematous. In this figure 8.4 you can easily see that there is ciliary congestion. Ciliary congestion means that all the vessels that are present or that radiate from the limbus would be congested, due to which there is ciliary congestion. Or we can define it as pinkish area around the limbus due to dilatation of vessels which appear to radiate from cornea, iris and ciliary body. And you can also see clearly here hypopaeon, as I told you before, hypopaeon. And then this is the active ulcer. Whenever you will give cornea a stain, that particular ulcerated area would get 2% fluorescent dye, and this is a pathognomic sign. Pathognomic sign is a sign that is particularly related to that a particular condition, and it is diagnostic sign, we can also say. Other signs you would see in your patient are iritis, as this iris it would be inflamed. This is ciliary body, it can also become inflamed. It is known as iridocyclitis. Why it occurs? Because the infiltrative cells when comes in the anterior chamber, they can cause the inflammation of these areas as well. Now, another sign you would see is raised intraocular pressure, that is known as glaucoma. But here secondary glaucoma occurs, it means glaucoma which occurs after bacterial corneal ulcer. Now, why it happens? Whenever infiltrative cells come in the anterior chamber, they block this iridocorneal angle, and in this angle, this particular aqueous humor that is present here, it is drained through canal of Schlemm and etc., whatever the process is. But whenever this iridocorneal angle is blocked, that canal is also blocked, aqueous humor drainage would not occur, and there would be raised intraocular pressure. So all these signs you would see in your patient. So clinical diagnostic points are pain, corneal stain positive, hazy cornea, ciliary congestion, hypopyon may be present, as we have talked about all of these things. Next comes management of corneal ulcer. Corneal ulcer is a potentially bleeding condition, blinding condition, so you must treat it as immediate as possible. First of all, you must decide that is the patient worth to administer him or her in the hospital. So if you want to admit him or her, then admit. Particularly you will admit the patient if he or she is one eye patient, or whenever there is severe infection and in non-compliance. Then after admitting the patient, you would identify what are the predisposing factors, that why this particular disease, why this particular bacterial corneal ulcer has occurred. Either he or she wears contact lens, either there was any previous ocular trauma or any ocular surface disease, or if the patient is immunosuppressed, it means diabetic or any AIDS patient or any other immunosuppressed patient, like older persons are also immunosuppressed. Then you would do investigations before starting antibacterial therapies. What are routine investigations? You would do blood and urine test. In blood test, you would do CP, ESR, sugar, and in urine you would do this test. Then comes microbial investigations. You would obtain the material from the base and margins of the ulcer. And how would you do this? You would first of all anesthetize the cornea topically, and then you would take Camora spatula or bent tip of 20 gauge hypodermic needle. Then what you would do with that scrapping, you would do gram staining and Gemsa staining for identification of the infecting organism. 10% KOH is used to rule out if there is any fungal hyphae, because now you don't know that either it is due to any fungi or any bacteria. Okay, not only on the basis of clinical features you will start treatment, you must have to rule out all the other possible conditions. Then you would do culture. In the next video we will talk about the treatment. Allah Hafiz.